again, chapter 11 is really everything about controlling microbial growth out in the environment, things we do up in our lab, and then chapter 12 we'll talk about on Tuesday, different antibiotics, how they all work, what's happening with overuse of antibiotics. In chapter 11, we're going to talk some terms when we're talking about different types of disinfectants. What are some considerations on choosing which method do we use? And then we break down control of microbial growth into physical methods and chemical methods. There are chemicals like bleach, which is different than physical methods like incinerating or autoclaving. So a couple terms, and no, I'm not going to hold you responsible, and it's open note anyways, for the eval. A couple terms I just want to point out because you hear these terms a lot, and there are differences between them. Anything that talks about, sterilization versus sanitization, or sanitization, it's like a horrible, sanitize. Sterilization really means it does kill everything. Like endospores, those hard to kill bacteria, it kills everything. If something is sterile, there is nothing growing whatsoever. And ultimately, that's what we want to do in our lab. Real life, it's hard to get there because it does take some really strong disinfectants to really sterilize something. And honestly, sometimes we just don't need to kill everything to try to stay as healthy as we need to. Instead, a lot of the term that you hear most often is sanitize. And sanitize means it kills most bacteria. And I hear this word all the time, and everyone always thinks, oh, I sanitized it. I killed everything. It's like, no, you didn't. Sanitize really means you killed most organisms, but it doesn't mean you killed everything. And so a lot of times the ultimate goal of places is I want to sanitize. Example, we sanitize things in restaurants. We use water to sanitize. We wiped it down. We sanitized it. Doesn't mean you killed everything. The idea is, again, you're hopefully killing most of the pathogenic bacteria, hopefully, but it doesn't mean you killed everything. So it's like just knowing those couple differences in those words. The same thing, disinfect kills most organisms. If you disinfect something, you're like, I disinfected it. Doesn't mean you killed everything. It's kind of the same type of definition as sanitize, except sanitize is usually used a little more broadly out in the, kind of as you go about day-to-day life. Disinfect is usually used more medical term-wise, but again, it doesn't mean you killed everything. If something's disinfected, there could still possibly be things growing. So those are just a couple of the terms that you hear and you will hear as you listen to commercials and things like that. You're like, it disinfected. Didn't kill everything. And I'm like, we sanitized it. Didn't kill everything. But the job is to kill most bacteria. So, the big question is, how do we kill microorganisms? There are different ways to kill microorganisms. Two of the biggest kind of groups of how to kill them is we're going to alter their outer cell wall or membrane. So we're going to use disinfectants, we're going to use antibiotics, we can use different ways that we can damage kind of their outer protective shell. Again, that cell wall gives it strength, gives it structure. The cell membrane helps regulate what things go in and out. And if you damage those, things get out that shouldn't get out, things get in that shouldn't get in. If you damage a cell wall, just like if you damage the wall of a building, the whole thing falls down. So we can target that outer membrane or the wall, or we can try to damage some proteins or nucleic acids like DNA. So anytime we damage a protein, it loses its shape, we denature it. And if you denature a protein, it can't do what it's supposed to do. And so enzymes that we talked about last time are proteins. If we damage those, it can't do what it needs to do. It can't do all those chemical processes. If you damage the DNA, it can't reproduce if it's got damaged DNA. It's a huge mutation. And so we're kind of trying to target some of, the outer structures or some of the proteins or nucleic acids that we find inside these bacteria cells. Now, how do we select the right antimicrobial agent? Which one should we grab, which one should we use, which one should we buy? Well, we like to have them, our ideal antimicrobial agent is cheap. We want things cheap. Businesses want things cheap. We want it to work really, really fast. I won't lie, there are some disinfectants, again, that doesn't mean they kill everything, that you have to spray on a surface and let it sit for 15 minutes. That's not usually something a restaurant would probably gravitate towards. It's like, here, I'm gonna spray it on a table, but no one can sit there for 15 minutes. I'm like, so we want things that work real fast if we can. We want them stable, especially at room temperature. We want to be able to store these things on a shelf. They're gonna stay active. They're still gonna keep working even a year from now. They're not gonna explode, because some things could. We want them to be nice and stable and hang out. And we want them harmless to humans. We want them harmless to animals, and we want them harmless to objects. We don't want to spray some type of disinfectant all over an object and it literally dissolves the object. That wouldn't be good. We wouldn't want it to damage or cause issues with humans and animals. I mean, that's a whole lawsuit all in itself. So we want them harmful to the organisms, but not to anything else. That's our ideal agent, our perfect world. Now, some factors that we try to consider before we grab whatever agent that we want to use is what site, what are we actually disinfecting? Are you disinfecting something that's metal? Are you disinfecting something that an animal or some other type of person is going to touch? Are we going to disinfect a metal that might rust? I'm like, what is the site that we're going to be treating? And are there any environmental conditions that we need to know about? Are we disinfecting something outside today that we need to make sure we work with something that can handle and work at minus whatever degree temperature? Or are we disinfecting something that's going to be a room temperature? My temperature changes can affect how well a disinfectant works. If there's any pH differences in the environment or surfaces or whatever it is that you're going to use, we have to take that into consideration. Different disinfectants work at different pHs as well. We also have to consider, what are we trying to kill? So there are different organisms that are more or less susceptible to disinfectants, meaning there are some organisms that are easily killed, like the simplest disinfectant. Just basic soap could kill them, or they dry up real fast on a table. There are some things like the bacteria that make endospores that are really hard to kill. And so we have different levels of disinfectants for what levels of organisms we're trying to kill. So an example of a high-level disinfectant. These are the things that are going to kill everything. A high-level disinfectant really should be something that's going to sterilize, meaning it's going to kill everything, including those endospore bacteria. So in your medical facilities, if you have a patient that has C. diff, that is an endospore bacteria, you're going to need to use a high-level disinfectant. Your basic Lysol spray is not going to kill it. You need something that's meant for endospores, which usually the bottle will say sporicidal, because it has that word spore in it. That's talking endospores. Something that's an intermediate level. So these, are harder to kill, but they're not endospore. Intermediate level disinfectants are going to kill fungus, they're going to kill protozoans, and they're going to kill most pathogenic bacteria, which usually means the gram-negatives. So they're going to kill quite a bit. They won't kill everything. And any intermediate level disinfectants, just an example, would be some of like your Lysol. Your Lysol is an intermediate level disinfectant. It's going to kill most things. It's not going to kill everything. A low-level disinfectant is going to kill it'll kill some pathogenic bacteria. It's going to kill a lot more of gram-positive bacteria. They're just more easily killed. It may kill some viruses. These are the most susceptible. So it may kill some viruses. Won't kill them all. An example would be a lot of your soaps. A lot of your soaps, they do get rid of a lot of things and inhibit the growth of a lot of things, but they're not going to get rid of everything. A lot of times washing your hands, it's a combination of using soap to kill some things and then just the physical act of rinsing bacteria off of your body. Like, you're just not killing them, but that soap is like releasing them from your skin. Oh, here's my little arrows. Now, as I said before, we do group our types of microbial growth into physical methods. We're going to physically kill them, and then chemical methods. What chemicals can we spray or use on different surfaces or areas to kill microbes? So physical methods, we can use heat. to kill organisms. Why do we use heat? Well, the effects of the high heat. When you heat up a protein, it will denature a protein. It's the same exact thing happens when you cook an egg. You denatured all the proteins. The cell membrane of a bacteria is made up of a lipid. If you heat a lipid, it softens it, so it's really damaging that outer cell membrane. So we can add heat to organisms to kill them. Now we do group heat and heat-related methods into two groups, moist heat and dry heat. So if you don't like the word moist, you're gonna hate today's lecture because I use the word moist a lot. So again, moist heat, it's using heat, anything with a little bit of water or moisture involved in it, which is more actually effective than dry heat. One example of moist heat is boiling. It's heat and water, moist heat. Now boiling does not kill everything. Again, before we even knew about bacteria, they did know that boiling, was what was used in hospital settings. And it does kill a lot, but it's not going to kill everything. Some of the things that it will kill is it will kill most bacteria, but it will not kill endospores. So it will kill most bacteria, but it will not kill endospores. So boiling, if you didn't have anything else, which they didn't way back when, boiling worked great. And I'm like, it killed most organisms. It decreased the possibility of infections with any types of medical procedures, but it didn't kill everything. So to help with that, we have what's known as autoclaving. Now, what temperature does water boil at in Celsius? You guys know? It's a nice even number. 100. Again, we should always just do everything Celsius. And in the medical field, you will. So water boils at 100 degrees Celsius. Again, that's not enough to kill everything. However, what we can do is we can autoclave it. We are going to add pressure. And so just like you pressure cook something, when you add pressure, it allows us to increase the temperature higher before it gets to a boiling point. And so what that allows us to do, it allows us to get that fluid up to 121 degrees Celsius. So it allows us to take it past boiling because we've added the pressure to it. If we can have that temperature at 121 degrees Celsius for 20 minutes, we can say we've sterilized things. So in the medical field, as you guys are all working, and I'm like, when you pull, any type of usable forceps, things like that are reused, they're usually autoclaved and then sealed up to stay sterile. And again, it's because we can increase that pressure. Do any of you guys have an Instapot? I don't. I haven't jumped on that bandwagon yet. But it's the same exact idea, is that you're adding the pressure, which allows you to up the temperature, allows you to cook things faster. We'll get into a whole discussion on canning foods. Does anyone can foods? Or have family members that can foods? There are two different ways you can can. You can boil can, and you can pressure can. And we'll talk about why you would choose one over the other later, when we talk about all the problems if you choose the wrong one. But that pressure canning does allow you to increase the temperature. It is actually better if you have a pressure canner. This is showing just an example. You can get huge autoclaves. This is actually just like the one we have upstairs right next to where you grab your lab coats. That big huge thing that is hissing and will beep and make loud noises. That's that pressure that you'll hear hissing and making some of those noises. And then it has a whole computer output because it is constantly monitoring that pressure and the temperature. And although I was like, it has to stay, it only takes 15 to 20 minutes to autoclave something, you have to realize it takes a long time to get up to that pressure, and it has to maintain that pressure for at least 15 to 20 minutes. So at any point that it gets below that temperature of 121 degrees, it stops the time till it gets back up there. And then it takes forever to cool and release all that pressure. So something that goes into an autoclave could easily be in there for 2 to 3 hours. So we have a huge one upstairs, but you can get small little tabletop ones, depending on what you're autoclaving. Now, to make sure that the autoclave actually worked, because everything that's in there should be killed, and you want to make sure that it maintains that pressure and that temperature for at least 15 to 20 minutes, they do usually put in some type of indicator. This is showing an example of like a little broken glass ampule that if it maintains the correct temperature for the correct amount of time, it will stay the same color. If it doesn't, then there'll be a change in color that would say that, nope, we didn't actually maintain that temperature for that length of time. These are kind of putsy. Instead, we actually have tape that you can buy. It looks just like masking tape. Like it looks exactly like masking tape when you buy it. You need to tear a little piece off, stick that masking tape on there, and there's actually something etched on the outside of the masking tape that will change colors if it maintains it. That will actually even say, it will like show up dark black by the next, when you pull it out, that'll say autoclaved. It's an indicator. It's just a chemical reaction that's happening on that tape, which is a lot cheaper and easier to use than these. Now, another example of moist heat beyond boiling and autoclaving is pasteurization. Now, the very first week, you guys had to do a little bit of, history of microbiology worksheet. There's Louis Pasteur, which is where it gets its name from. Pasteurization is used for usually food and beverages. Now, it was initially used for keeping wine and beer from spoiling. It was great. Wine and beer didn't go bad once it was pasteurized. So it's usually used for some type of beverages and different types of foods. So a lot of times we'll use it for milk. That's usually where you hear it the most. We use it for juices. We even use it for some yogurts. Again, you guys will see yogurts. It doesn't kill everything, but it kills a lot. And so it's not a sterilization method. There are still some organizations that can survive. However, we've taken pasteurization. Again, your milk is pasteurized. Most organisms are killed. However, milk itself, even when refrigerated, and refrigeration's a whole nother method too, can still survive. This is why milk has a shelf life of usually about a week or 2 weeks. There easily are microbes still in your milk. So there are things we can do though, is we can take pasteurization and we can up it. And it's called ultra high temperature sterilization. It is pasteurization on steroids. Again, pasteurization, it does not get to a temperature that it's going to kill everything. If you boil milk, it will, if you get milk at a certain temperature for long enough for it to slowly warm up to boiling, there's protein in your milk. And that high temperature changes the proteins, which means boiled milk will not taste the exact same as cold milk. And we don't always want that. That's why we don't boil our milk to pasteurize it. We don't get up to that temperature for that slow. But we have methods that we can get that milk up to 140 degrees Celsius, and we can do it very, very quickly. It's huge machines that do it, to get it up to 140 degrees Celsius for just 1 to 3 seconds. That temperature is higher than autoclaving, so it is under an extremely high pressure, and it's almost like a flash boil. And as long as it can get up to that temperature for that short amount of time, it's not a long enough time to change the proteins all that much. So milk stays more tasting like your normal milk you're used to. And you can actually store milk at room temperature. Have you guys ever bought or used milk that's bought right off the shelf? Not in a refrigerator? And you're like, oh my God, I would never. It's super common, not always here, but in other parts of the world. There are parts of the world that they don't have refrigerators in their houses, which means even once you open the milk, they can't refrigerate it. And so they have to buy something that can be stored on the shelves. Granted, once you open these, you expose them to possible microorganisms, and refrigerating is best. But you can buy room temperature milk at the store. If you go to the grocery store, look around for it. It's like usually it's in like little tiny containers, a little almost like a cardboardy type container. But you can buy milk right off the shelf. Again, it's not common around here because everyone's like, oh yeah, it's so gross. If you didn't have a choice, it's what you could buy. Now we're gonna switch away from moist heat, because I just used the word one more time, and switch to dry heat. So we use dry heat on materials that can't get wet. An example is a lot of metal utensils. If you get metal utensils wet, what can happen to them over time? They'll start to rust. And so even if we needed to disinfect certain like powders, things like that, we can't use moist heat. But you can use dry heat. Now, dry heat is not as effective as moist heat, and so to make it as effective, it requires a higher temperature for a longer period of time. A temperature of 100 degrees Celsius without that water as well is not going to kill as much. And so one of the easiest ways we can use dry heat is incineration. This is why you guys use this in the lab upstairs. And I'm like, that is an extremely high temperature. I know it's written up there somewhere too, but I think it is like 1,500 degrees those things can get up to. Higher temperature than 100. And I'm like, it's not usually for a longer time, only because ours are so high in temperature. But if you had them at a lower temperature, it would require a longer time at temperatures. They're just not effective as moist heat. Some other physical methods, flipping away from heat and flipping the other direction, is refrigeration. Now, refrigeration and even freezing will not kill bacteria. It will just slow their growth. So if you put foods and things in the freezer and you're like, this'll kill them, it won't. I mean, a long enough time it may kill some, but it's not going to make your food not have bacteria in it. So all it does is when you decrease the temperature, that lower temperature just decreases all of their chemical reactions. It decreases their growth. It decreases their reproduction. It doesn't stop it. It is why we have a refrigerator in our lab, because we can pull those plates right out of the incubator. We can put them in the refrigerator, and your bacteria are still alive. And I'm like, we've just slowed their growth that they're not really doing a whole lot of just hanging out. Now we could freeze our bacteria as well, and there are two methods to freeze it, and they have their differences depending on what you want to do. We can slowly freeze things, and we can do quick freezes as well. Kind of depends on whether you want to just keep the bacteria alive, for further use, or whether you want to more kill them. Slower freezing, so even just putting it in your freezer at home, it takes a while for it to slowly cool down. You think about how long it takes for ice to form in your freezer. When you slowly freeze things, it gives water enough time to form ice crystals, which means if there's water inside your bacteria, and there is, it could form ice crystals inside your bacteria, which could damage the bacteria to kill it. And so, not all of them. Again, freezing will not kill bacteria, but it will kill more of them. And so putting bacteria in a freezer like you have at home that is freezing temperature, but it's really not that cold, is more likely to kill some of the bacteria just because of the ice crystals that can form. Quick freezing is when you use an even colder temperature, which means the water doesn't have as long of a time to form ice crystals. The bacteria will survive for use. And this is what we do up on our lab. Again, the bacteria we work with this semester that Arlene makes, these bacteria are kept in freezers until we need them, which means we want to keep them alive. And so we actually have a minus 80 degrees Celsius freezer upstairs. It has to be this low because it allows those bacteria to quickly freeze without those ice crystals forming, and it doesn't kill the bacteria. So we can use them, we can grow them, we can study them later on. So it kind of depends on, what are you trying to do? Do you want the bacteria to more die, or do you want to keep them alive like we do upstairs? Desiccation just means you are drying them out. And it's one of the easiest ways to kill a lot or inhibit a lot of bacteria growth. Bacteria are not going to thrive on this table up here. It's dry. That doesn't mean there aren't some bacteria hanging out for a little while. But if I walked away, and I'm like, unless they're endospore, I'm like, they're not going to survive pretty much for 24 hours or longer. Removing water stops bacteria from growing. If it can make an endospore, it will go into an endospore state, but not all bacteria can do that. It is one of the ways, before we knew bacteria, on how we preserved meats. We dried them out. It's kind of like the beef jerky. We realized if we removed all the water, that meat stays good a lot longer because bacteria need water for survival. This is even why, an apple It's going to go bad after, I don't know, a few weeks. But if you dry an apple in like a dehydrator, you have little apple slices, that lasts a lot longer. Lyophilization is the word for freeze drying it. So you're removing water and freezing it. If you remove the water before freezing it, you reduce the chance of any ice crystals from forming it. And it's how usually like the CDC will maintain organisms by freeze drying them. Just we've got stall cultures. They last a really long time. So it's not killing them, it's just removing the water, which you can rehydrate them later, and it's not damaging them because they don't have the water to form ice crystals. We don't do that upstairs though. Another way to inhibit growth is to remove them through some type of filtration method. So if any of you guys have like a Brita filter, things like that, the idea is that larger things are caught on the filter, which decreases the chance of any microbes being in or on whatever surface it is that you want. So hopefully the filter is going to have holes in it to allow a fluid to go through, but not large enough that bacteria can go through. Viruses will always be able to go through those kind of filters though. Even some bacteria are very small and can go through some of those filters. The idea is that you're just trying to remove those bacteria from that fluid, or if we don't even need filters for fluids, if you want to remove filters from the air. We do have air filters. A lot of times your air conditioners, your furnaces have air filters on them. The idea is that removes some microorganisms, not all, removes them from the air. It traps them. Some filters are better than the other. When you start to see the word HEPA filter, there are usually, the filters themselves have charged, it's almost like a charged filter, and then smaller organisms are attracted to those charges and they stick. Which means it removes it more from the air. Our hoods, we have a couple hoods upstairs in our lab. Usually Arlene uses the one in her stock room, but they do have HEPA filters in there. So any air that's anywhere near where we're working with microorganisms, the air goes through a HEPA filter to decrease the possibility of microbes landing on anything she's working with. Another physical filter is osmotic pressure. And this does work a little bit with not quite desiccation, but kind of along those same lines. If you have a high concentration of salt or sugar, water wants to go where there's not as much water. And so if you add a lot of salt or you add a lot of sugar to things, and I'm like, that's where water is going to want to go. But it also means that if you have a lot of salt or sugar, that salt or sugar will pull water out of where bacteria need it. So the salt and sugar pull the water from an area that bacteria want to grow, which means they don't have the water available for use for them. So it's a little bit of the desiccation because it's pulling water from where the bacteria are growing, and that water is going to wherever that salt and sugar is. So cells that are placed into a hypertonic solution. Now the hypertonic, that's when they have lots of solutes, very little water. It means hyper means they have a lot of something. So if they had a lot of sugar or a lot of salt and you put a cell in it, that cell's going to lose all the water because the water's going to want to go where that salt is. The cells could shrivel up. This means it could be bacteria as well. This is why some of the best ways and oldest methods of preserving food is to add a lot of salt and add a lot of sugar. This is why ham, which usually has a lot of contaminants in it, is like super salted. Your beef jerky, we've removed the water. Oh, we also add a ton of salt into it. Using salt and sugar maintains foods for longer periods of time. Again, it's because that substance is removing the water from the bacteria. However, fungi can withstand the loss of water more than bacteria. And so if you have foods that have a lot of salt or sugar in them, generally you don't have a lot of bacteria growing in there. However, you usually have a lot of fungi growing in it. A big example of where you might see this in your refrigerator. Again, refrigerator decreases the temperature, not as much grows. Some bacteria will grow just fine in the refrigerator though. We'll talk later about some examples. And fungus can grow just as fine in your refrigerator. But if you had like a bowl of, or a container of jelly, so in the refrigerator, jelly's got a lot of sugar in it. That's going to inhibit bacteria growth. Because it's in a refrigerator, it's going to inhibit bacterial growth. However, fungus will grow. And so it's one of the things that if you pull stuff out of your refrigerator, it's not bacteria that's causing fuzzies all over your stuff. It's fungus, because fungus can handle low temperatures and they can handle high sugar areas or high salt areas more than bacteria can. Another physical method that we can use to stop bacterial growth is using radiation. Now, both of these, whether it's ionizing or non-ionizing radiation, the radiation is going to, the ions, the charged particles that's in the radiation is going to damage DNA. But we do group the types of radiation into ionizing and non-ionizing. Ionizing, the wavelengths are shorter and it is more likely to be able to penetrate into the surface of something, like so inside. They're just smaller wavelengths. They can get inside of molecules or substances better. Non-ionizing radiation, their wavelengths are greater, and they're used for disinfecting surfaces because those ions cannot penetrate, inside of something. The most common example of a non-ionizing radiation that's used all the time is UV light. UV light is used to sterilize surfaces. If you ever walk into Walmart by the carts, you'll look now, even above the carts, they actually have a UV light that hangs out by the carts. The idea is it sterilizes the surfaces of the carts. Although you can have wipes now and wipe those. That was added with COVID. But otherwise, UV light is great at sterilizing surfaces, but UV light is not going to penetrate inside of something. Now, most common types, we use radiation for sterilizing other than UV light to sterilize surfaces. They use radiation quite often with different foods. Radiation, because it can kill microorganisms without damaging the inside structure of the food itself. If we use some non-ionizing radiation on fruits and vegetables, this is showing raspberries that were exposed to radiation after 5 days. This is showing what happens to them if they weren't exposed to radiation. So we do actually shoot some UV light and radiation at different foods because it's going to kill all the bacteria and even some of the fungi that would normally break those foods down. So your foods last longer. If you never realize that they do that to your food, they do. Now, when we work with different organisms, we do talk about them, are they do we need a high-level disinfectant, low-level disinfectant, depending kind of a little bit on how hard they are to kill. But we also group our organisms, not just on how hard they are to kill, but we group organisms we work with in the lab or in medical facilities, kind of how likely are they to cause disease. And so we'll talk about which ones we work with. I thought I had another picture. There we go. We group organisms into biosafety levels. So a biosafety level organism 1 does not cause disease in a healthy human, which means it's not going to cause disease in you guys. I'm under the assumption you guys all have healthy working immune systems. If you don't, let me know, because we should probably work on a few things in lab. But you guys all have healthy immune systems. You're not immunocompromised. And so these are organisms that would not affect you. So even things, when we talk about, if something's sanitized, well, we've killed a lot of organisms, but it didn't kill everything, but hopefully those other things that it didn't kill, you're healthy, it's not going to cause you any issues whatsoever. Almost all of the organisms we work with up in our lab are a biosafety level number 1. You guys are all healthy and they're not going to cause any issues whatsoever. I put an asterisk because we do work with a couple organisms that are in our lab that are called biosafety level number 1, which means they're moderately hazardous, which means they could cause disease in a healthy individual. Now, none of the ones we work with in our lab that could cause disease are going to kill you, even, especially if you're all healthy. But an example of something that would be considered a biosafety level number 2, we do work with the bacteria that causes strep throat. Even if you're healthy, you can get strep throat. And so we work with that bacteria. It's not going to kill you, but you could get strep if you licked the plate, which you're not going to do. So we do work with a couple organisms that are in that group. Biosafety level number 3, these are organisms that will always cause disease, even in the healthiest individual. I don't work with any of these guys. Examples, I want to say like C. diff, but honestly, I'm like, that usually you have to have another issue going on as well. It's going to cause disease, even in the healthiest individual. We don't work with them. And biosafety level number 4, not only will they affect anyone, doesn't matter their immune system status, they will cause severe and or fatal diseases. An example, I had a picture, there we go. Ebola, which we'll talk about when we talk about viruses, is a biosafety level number 4. That if you're working with something at that level, I mean, you've got the full hazmat, you have no exposure to oxygen so that you can't possibly breathe anything in. You've got your own oxygen supply because this is an organism that will cause disease, even in the healthiest individual. It's a very severe disease, if not fatal. So, no, we would never work with anything like that. Now, we're going to flip it from physical methods of killing to chemical methods of killing. So we're going to add some type of chemical. Now, things that they can affect. So if we use different chemicals, we want those chemicals to affect a microbe's cell wall, cell membrane. These are the things I had on that one of those earlier slides. So their cell wall, their cell membrane. We could have chemicals that work on proteins or DNA. And I got to put in there, some organisms have RNA, or their RNA, their nucleic acids. So different chemicals target different areas. Now, chemicals will work in different environmental conditions. Again, as I said before, we do always have to know what is the temperature of the area that we're trying to disinfect. You know, like, is it super hot? Is it super cold like outside today? And I'm like, it's going to affect how well the different chemicals work. Is there, any kind of pH changes that we have to be worried about? Like, just what are some of the other environmental conditions? But we have chemicals that are effective against all microorganisms. Just depends on which chemical you grab. So we do have chemical methods that are effective against all microorganisms. This is when we didn't have lecture. Ignore my last little thing. We're going to go over it, but I'm going to do a review. I know most of you guys are going into nursing. Not all, but most of you guys are going into nursing. So I know you're going to have to take a pharmacology class. Here is hard because it's a lot of memorizing. And I'm like, and a lot of the chemicals are going to be talked about in there, and a lot of the antibiotics that we're going to go through on Tuesday are going to be on there, which is why it's open note. You guys don't have to know them all yet. At some point you will. But some examples of different chemicals that we can use, and then a little bit on how do they work. So phenols and phenolics are intermediate to low level. So these are not going to be those high level disinfectants meant to kill everything. Their job is they denature proteins and they disrupt the cell membrane. That's how they kill their organisms. And they are commonly used in healthcare settings, in labs, and in homes. One of the most common example, Lysol. Again, it's not going to kill everything. It's not going to kill C. diff. But if you're not suspecting a patient has some, really hard to kill organism, they're quite cheap. They're easy to use. You spray down, wipe it down. And it kills most organisms. Another one that's commonly used are alcohols. They're intermediate level disinfectants. Again, they're going to kill a lot of organisms, not all. Their job, they denature proteins and they disrupt those cell membranes. And they have done enough studies. Alcohol, even your basic hand sanitizer, is more effective than soap at removing or killing bacteria from your hands. So soap's job really is to help facilitate the removal of the bacteria. but the soap itself generally doesn't kill bacteria. That's what that alcohol's there for. And why before any type of shot or injection, they wipe the surface of your skin with an alcohol wipe. It's going to kill quite a bit of bacteria. It is an intermediate level, but it's not going to kill everything. Halogens are intermediate level organisms. They target enzymes, which again are proteins. Examples, and this is where I got a bunch of pictures of halogens. Iodine, that's used. I don't know if you've ever had to travel somewhere where water's sketchy. You can actually get iodine tablets to put in your water. That's going to inhibit bacterial growth, and you can drink low levels of iodine. It's just fine. More iodine. Chlorine that they put in your pools are halogens. Some of your bleaches, halogens. They actually chlorinate your drinking water. La Crosse chlorinates their drinking water. Sometimes you can almost smell the chlorine coming from the water. I swear sometimes it's higher levels than not. And bromine, which also a lot of times is used in pools, inhibits a lot of bacterial growth. Again, they're intermediate levels. They're not going to kill everything. And their job is just to target those enzymes. If bacteria don't have enzymes that can't do other metabolic reactions, they're going to die. Hopefully. Oh, there's more chlorine. Another iodine picture. We have oxidizing agents. This would be an example of a high-level disinfectant. So this is where we're going to get into more of a sterilization method. They kill by oxidizing enzymes. And again, it's where changing up compounds. Oxidation is loss of electrons. Common examples of oxidizing agents, these are going to be the ones you would have to grab if you would suspect C. diff. Hydrogen peroxide. I have pictures, there you go. Ozone and peracetic acid. Ozone, so your normal, this is oxygen, it's O2. Ozone is actually O3. And they actually use ozone in La Crosse's drinking water. I was super fortunate to be able to tour the water facility there. And before any of the water leaves the water facility, they actually ozonate the water. It's a high-level disinfectant. Your La Crosse water, as they say, a lot of times, city water is cleaner, healthier than bottled water, because bottled water doesn't always go through ozonation. They do like a reverse osmosis. Surfactants. These are low-level disinfectants. This would be where you would find your soaps. All the job of a surfactant is to almost loosen the bacteria off the surface so that it falls off. So a soap, its job is to loosen that bacteria. It can't stick as well to your hands, and so it should wash off with water. So they reduce surface tension. That's what allows things to stick to each other and stick to surfaces. And they are more likely to get washed off. But they're low level. So a lot of times what they do with different types of soaps and detergent is they will add something else into the soap, like some type of alcohol or some other type of disinfectant, so that it not only physically removes the organism, but it also has some type of bacterial killer in it. So it's all about reducing the membrane. They just can't stick to each other. They can't stick to surfaces, which means they're just going to fall off with the water. Heavy metals. Heavy metals inhibit bacteria, but they're a huge inhibitor of fungus. Again, fungus, they're pretty resilient. They can grow in refrigerators. They can grow in different environments bacteria can't. And so we use heavy metal in different areas. Their job, denature proteins. Couple examples, silver nitrate, thimerosal, which, has anyone heard of thimerosal before? This was like a huge, thimerosal was in the early, was the early 90s, late 90s, when there was that one study that someone put out and said, vaccines cause autism because there's thimerosal in it, which is a heavy metal. They did use, in some of the initial vaccines, they did use thimerosal. Again, vaccines, we want those stored at room temperature. We want those easily stable. And we did have things that preserved the vaccine so that you wouldn't get bacteria or fungus growing in it. And so they're like, oh, nope, it caused autism. One, he had no results that actually proved that. Actually, more people didn't get autism that got vaccinated than did. And they've removed thimerosal from vaccine like 20 years ago. So it's never an issue. They're like, it never caused it, but we'll remove it to make everyone happy. But I still will see, because I'm like, I'm always going to have at least, one or couple of anti-vaxxers. And they're still like, oh, thimerosal causes vaccines. Why would you get your kids vaccinated? There's thimerosal in it. Prove to me a vaccine that has thimerosal in it. 20 years ago, they removed it to make all those anti-vaxxers happy. And they still think it's in there. Again, don't always believe everything you read on the internet. Copper, this one even is showing gold and silver will inhibit microbial growth and fungus. So there are certain areas or certain things that they do put heavy metals in to reduce growth, especially if they suspect fungus. Aldehydes usually means it's anything that's going to end in hide. These are high-level disinfectants. Their job really is to kill everything. Examples, glutaraldehyde, formaldehyde. Now I wrote on here formalin. Formaldehyde was used very commonly to preserve organisms. So you probably all had one of those science teachers at some point that had the jars of preserved organisms. Those are preserved in formaldehydes or in glutaraldehydes, and now formalin. And it's a high-level disinfectant. So those organisms, whatever is in those jars, they can sit on the shelf for 20, 30, 40, 50, 60 years. Nothing's going to break them down because they're in such a high-level disinfectant. Nothing's growing, which allows those organisms to be preserved in the fluid. They did eventually find out that formaldehyde caused cancer. So they were able to modify formaldehyde so that it's no longer carcinogenic, and it's now called formalin. So it's the safer version of formaldehyde. Gaseous agents, there are some things that you just can't put fluids on. It doesn't matter what, you just can't put a fluid on it, or you can't get that fluid to get to a certain area that we can use gas to kill organisms. An example, I've got examples on here. There are some things like different types of plastics that if you put some of these disinfectants on it, they will literally eat apart plastic. There are long tubing. If you've all seen patients, they got long tubes hooked up to things. How do you know the inside of that tube is sterile? We can sterilize those tubes in gas, because that gas can get all the way through that tube. Sometimes the disinfectants can't, and then you can't get them all back out. And so we do have gaseous agents for high-level disinfectants. So we use them on different equipment. But the disadvantage, they are hazardous, they are explosive, they are poisonous, and some of it's carcinogenic. So it's, you have to have highly trained people that can work with those. There are some enzymes we can use that can inhibit microbial growth. Again, one example, lysozyme is an enzyme that inhibits bacteria. We actually make enzyme in our tears, if you didn't realize. It actually inhibits bacterial growth because it digests that cell wall, the peptidoglycan. So lysozyme we make in tears. Prionzymes can actually damage prions, which we'll talk later about. But it's just an enzyme that can target the microbes, and it usually is targeting some particular structure like a cell wall or cell membrane. One more slide. And then my last slide I have is, we just call these as generic words, is an antimicrobial. So anything that inhibits bacteria of any kind, and a lot of what we just went over are antimicrobials. And so a lot of times you hear antimicrobial as just a generic term. All of these things are antimicrobial. And we use antimicrobials to treat and or prevent disease. But they're all used outside of the body, because it's the next chapter we go through on Tuesday. That's all the stuff that gets ingested, like your antibiotics. The only problem with antimicrobial use, doesn't matter whether it's some type of chemical or not, or if it's an antibiotic that we'll talk about on Tuesday, the more we use different antimicrobial agents, bacteria reproduce faster, which means they can adapt faster than we ever could. And antibacteria are becoming resistant to just about any kind of disinfectant or antimicrobial agent that we have.